Hey guys, and welcome back to my YouTube channel. So today we are going to be doing the carbohydrates, so part 2 of my sort of slideshow on carbohydrates. So make sure to check out my part 1 if you want to see that one before clicking onto this. Or if you just want to click onto you just want to see this, and that's fine, you do you. So we are going to be looking at polysaccharides, and this is AS and A level biology for all what's it called, all examples. That's it, for all examples. And also make sure to like and subscribe if you like this video and you want to watch more videos like this, because that would be awesome. So let's get started. Polysaccharides. So from my previous video on carbohydrates and disaccharides, polysaccharides are polymers of monosaccharides. So they are repeating units, they contain repeating units of monosaccharides. Examples of monosaccharides include glycogen in animals, starch in plants, which consists of amylose and amylopectin, and also cellulose in plants. Starch properties and functions. So starch is used as an energy storage, and it is found in photosynthesising cells such as palisade cells in the leaves, and storage cells in the seeds and storage organs. So it is compact, so that there's like a lot of glucose molecules packed in a small amount of space, and they are insoluble, so it does not affect the water potential in the cell, therefore osmosis, and that is really good. And it also contain branches which can be easily broken down so that glucose can be used in respiration. So amylose is a long chain of alpha glucose molecules joined by a 1 to 4 glycosidic bond. It coils into a helix shape with hydrogen bonds that kind of gives it its shape, maintains the shape of keeps it in place. Yeah, keeps the helix shape in place. The hydroxyl group on carbon 2 are located inside the coil. This is kind of extra information, which makes the molecule more insoluble and allows the hydrogen bonds to form and maintain its shape. Yeah. Amylose. So as amylose molecules has two ends that an enzyme has two because amylose has two ends and an enzyme can attach to, it means that amylose is broken down more slowly. So what I mean by that is, if you can see from the picture on the slide, you can see that at the top there's an end where a enzyme can attach to, and at the bottom there's an end that the enzyme can attach to. So these two ends are the only ends that an enzyme can attach to, and because it's only two ends that they can attach to, that means that breaking down amylose to form like a monosaccharide, which is alpha glucose, it's gonna take a longer process. So that's what it literally just means. Amylopectin. So this is a long chain of alpha glucose molecules joined by a 1 to 4 glycosidic bonds. It coils into a helix shape, which are held together by hydrogen bonds. However, compared to amylose, amylopectin, they actually have branches, and these form a 1 to 6 glycosidic bonds. So the fact that there are branches makes it more accessible for enzymes to attach and bind to, and as a result it means that amylopectin is broken down quicker than amylose, as you can see here. So there are three places in this photo that enzymes can attach to, therefore the rate of hydrolysis is faster. Sorry. Glycogen. Properties and functions. So glycogen is used as an energy storage and it is found in liver cells and muscle cells because they have a high metabolic rate. It is compact, so there is a lot of glucose molecules packed in a small space, and it is insoluble, which is really good because it does not affect the water potential in the cell, therefore osmosis. And the branches, it also contains branches which can be easily broken down so that glucose can be used in respiration. So glycogen has a similar structure to amylopectin, and it is made up of alpha glucose molecules that join with a 1 to 4 and 1 to 6 glycosidic bonds. In glycogen, there are more 1 to 6 glycosidic bonds, so it's highly branched. And the fact that it is highly branched means that it is even more accessible for enzymes to bind to or attach to. Another way of saying this is that it provides a larger surface area for enzymes to attach to. So that's also another saying that you can also say. Glycogen that means that glycogen is quickly hydrolyzed, broken down by enzymes for respiration. So this is very important because animals, animals and humans have a higher demand for glucose than plants because of its high metabolic demand. We are moving more. For an example, maybe an animal wants to run away from its predator, then they need lots of glycogen, lots of glucose. So the fact that glycogen is highly branched means that enzymes can quickly hydrolyze and break down the glucose molecules that are attached and break the glycosidic bonds, and it just basically makes it faster. So we humans and animals have a higher demand for glucose than plants. Plants, they don't really move. I won't say they don't move. Most plants are stable, like they are not mobile. Yeah. And yeah, as I said, this is because animals move around, which requires muscle and so on and so forth. Cellulose, functions and properties. So they are structural carbohydrates in plant cell walls. Cellulose is strong, which prevents the plant cell from bursting when there is an excess of water molecules entering the cell by osmosis, and it makes the plant cell turgid. Cellulose is made of long, straight chains of beta-glucose molecules joined by a 1 to 4 glycosidic bond. And in order to form this 1 to 4 glycosidic bond with beta-glucose molecules, every other beta-glucose molecule must be rotated 180 degrees. So let's say you have this beta-glucose molecules, and you have another one. They can't form a 1 to 4 glycosidic bond like this. So this one needs to go 180 degrees for it to form, basically, to form a glycosidic bond. And as a result of this, it prevents the chain from spiraling compared to amylose. It doesn't spiral. And there are hydrogen bonds between the rotated beta-glucose molecules in each chain. So this gives an extra strength and also prevents spiraling. Cellulose, they are the straight chain of the beta-glucose molecules run parallel to each other. So you have one chain of beta-glucose molecules here, and then you can have another chain of beta glucose molecules here, and they're running parallel to each other. And yeah, there are hydrogen bonds between the adjacent chains, which provides additional strength. So basically, this is a chain of beta glucose, and then this is a chain of beta glucose, another chain. And basically what I'm saying is that between this there are hydrogen bonds which are keeping it in place, and that provides additional strength. And even though like a hydrogen bond is seen as weak, but if there were thousands and hundreds and so many of them, that's what makes it stronger, the hydrogen bond stronger. So the fact that cellulose has thousands of many hydrogen bonds means that cellulose is stronger. Fibres of cellulose. So these chains of beta glucose molecules can form into a strong fibres. The cellulose chains can pack together to form microfibrils, and then bundles of microfibrils can then form a macrofibril. That should be macro, not micro. I'm sorry, guys. So basically, again, I'm just going to use my hand for this. So basically, these two chains, let's say there are so many chains, this would form like a microfibril, and then this, and then this, and then more of that will form a macrofibril. That should be macro, not micro. Sorry, guys. Cellulose. Unlike starch and glycogen, cellulose is hard to break down by hydrolysis, and this is because most animals do not have the right enzyme, which is cellulase, which can actually break down cellulose. And by breaking down cellulose, you mean like breaking the 1 to 4 glycosidic bonds between the beta glucose molecules. So most animals do not have that enzyme, do not have cellulase enzyme, which is used to break down cellulose. Herbivores such as cows, who have symbiotic bacteria lives in the gut, so in the stomach, and these symbiotic bacteria produces cellulase enzyme. There is a mutualistic relationship. Oh, that literally just reminds me of GCSE biology. I remember learning that. Because the bacteria will benefit as it has a habitat to live in, so it lives in the stomach, and the herbivores such as the cow will benefit because the cellulose would be broken down into glucose, which can then be used in respiration or so on and so forth. So there's a mutualistic relationship between herbivores and symbiotic bacteria. That's why some bacteria are actually good for you. So not all bacteria is bad or bad. Humans like us, we do need cellulose because cellulose provides the fibre in the human diet, and it helps keep the digestive system healthy. Even though we do not have cellulase enzymes, cellulose is still important for us. So we still need to, have that in our diet. Thank you guys for watching this video, and I hope you guys really enjoy it and found it good and informative. And as I said in my as I have said many times, make sure to like and subscribe if you like this video and you want more videos like this, because I would appreciate that a lot. And as always, I hope you guys have a great day, and see you in my next video, which is probably going to be the next day. So see you till then. Bye.